Hello, everyone. Can you hear me? You can. Apparently, you can hear me. I can see Amber and Fabian. Welcome, everyone, to the first RSVNet webinar that we are organizing to share new information with stakeholders in the field of RSV. It's the first tryout, and I'm very happy to see that many of you have registered for this meeting. I see lots of friends and good colleagues in the meeting, so thank you for being here. This webinar has been organized by the RSVNet Foundation. It is supported by Sophie Mazur and Amber Dasse and Leila Krachten from the foundation, who did a fantastic job in organizing this. And since it's the first webinar, you may see some technical challenges. I have learned that there is a delay in my voice. We are going to fix that for the next webinar. But you have to do, this is all we can do now. Now I'm going to switch to the next slide. Let me see. So our vision is to connect global expertise and leadership in order to decrease the global burden of RSV infection, using our mission to support performance of high-quality research to improve knowledge of RSV epidemiology and to develop safe and effective therapeutic and preventive interventions, with the goals to advocate and create awareness for the RSV infected children, to act as a focal point for effective partnerships with stakeholders, and to combine knowledge and capacity required to enhance development of novel RSV therapeutics. It's probably the last bullet point here that you're here for today. So why do we do RSV research and education? I think it is obvious, but we can't say enough that RSV is a major cause of burden in the world. 33 million patients with 3 million hospitalizations around the world in children under 5, with at least 120,000 deaths, most of them in the first 5 months of life. 99% of the children dying from RSV infection are dying in low and middle income countries. So we have decided that webinars may fill a gap in knowledge exchange, and our objectives for the webinar series is to promote knowledge sharing of the latest developments and publications on groundbreaking studies, and I hope today you will see an example of that. We aim to provide education for professionals, PhD students in the field of RSV infection, but also for the higher end of the professional researchers in the world. You'll see that today. Our ambition is to do that at least every 2 months. And the first webinar today has the title, What We Can Learn, How to Design and Make Our Own Vaccine Protein, with our speaker, Fabian Sesterhenn from the Institute of Bioengineering in Lausanne. The next webinar, please take your pen and write it down. November 19, we will have a co-host, a famous researcher in the field of RSV that will co-host with me. And the topic is still, it's almost finalized, but I can't share it yet with you. So there is, for this meeting, we have some practical points for you. You can ask your questions online through the little balloons that you can see here. Please ask your questions, and we will have enough time later on to ask Fabian all your questions. Please note your feedback for us. There is an evaluation form that you will receive after the webinar. Your feedback is valuable. This is the first time that we're organizing this, and anything is very welcome. Please help us enhance the quality of these webinars. And at the website, there will be the possibility to review it, to replay the webinar for your colleagues if they are interested. So with these words of introduction, I would like to introduce Fabian Sesterhenn. He is from the University of Freiburg in Germany. He got a master in molecular medicine, where he finalized his master's studies at the Scripps Institute in San Diego. And he was actually the first graduate student of Bruno Correa, at the EFPL lab in Lausanne. And last year, 2019, he obtained his PhD in bioengineering strategies to develop new vaccines. And I'm sure you're going to see a very, very interesting example of that today. Fabian, the floor is yours.
Thank you so much. Thank you so much, Louis, for the introduction. And thank you so much also to the RSVNet Foundation for organizing this seminar. And I'm happy that so many people are connected. So I'm very excited to tell you today about the work that we've been doing at the EPFL, where we combined de novo protein design to induce very precisely controlled RSV neutralizing antibody responses. So this article, which we published in Science a few months ago, was selected by the RSVNet Foundation as Article of the Month, and that's one reason why I'm here today. So just very briefly, we're located at the EPFL in Lausanne in Switzerland. So this campus is very beautifully located right at the Lake Geneva. And there, as Louis already pointed out, I got my PhD in bioengineering last year in the Laboratory of Protein Design and Immunoengineering that was founded just 5 years ago by Bruno Correa. And in this lab, what we're doing is really to combine protein design, which you will see a bit, to apply it to various immunoengineering problems. And here at the EPFL, we're affiliated to both the Institute of Bioengineering and also the Global Health Institute. So I guess RSV doesn't need a big introduction in this kind of audience. Nevertheless, I want to bring everybody on the same page. So RSV is really the leading cause for respiratory disease, especially in infants, and is also a problem in the elderly. So just a bit about the structure of RSV. So on the surface of this viral protein is the so-called fusion protein. So the fusion protein is essential for infection because it mediates the viral host cell membrane fusion. And this protein exists in a pre-fusion conformation seen here in the middle, and then a post-fusion conformation seen on the right. Now, what people found out over many decades is that, in fact, the pre-fusion conformation is what is most sensitive to antibody-mediated neutralization. And more so, there are particular epitopes that are present in the pre-fusion conformation, and some of them also in the post-fusion conformation, but most of them in the pre-fusion conformation, which are very sensitive to antibody-mediated neutralization. And perhaps most famously, and this is also one target I will talk about today, is the antigenic site 0, which is located at the apex of the pre-fusion protein and is targeted by really potent neutralizing antibodies. So just to give this a bit of a broader scope, so focusing antibody responses on particular epitopes is not only a challenge and of interest for RSV, but it's also for other pathogens. And so what I'm showing you here is the example of influenza, where you have a very variable, so sequence variable head domain in the influenza hemagglutinin protein, and a more sequence conserved stem domain. Unfortunately, what happens when we get seasonally infected with influenza is that antibodies that the immune system generates mostly target the head domain. However, the goal for vaccine development would be to actually develop immunogens which would elicit antibodies against the more conserved stem region here. So we as structural biologists have basically developed 4 different strategies, and we, in terms of the entire community, we have summarized this structure-based immunogen design strategy in a review that we published 2 years ago I will not go through the details of all these strategies, but instead I want to focus on the one that we are mostly working on, which is the idea of epitope scaffolding. So what epitope scaffolding means is shown here. So again, I'm showing you the RS virus, and then on the surface of this virus is the fusion protein here shown in the pre-fusion conformation. Then highlighted in red is the epitope that is targeted by the clinically approved antibody palivizumab. And so what was done in a couple of years ago by Bruno Correa and Bill Schief and published in 2014 is that they isolated this epitope from the viral protein and then transplanted it onto a computationally designed protein. And through computational design, they could design this epitope-focused immunogen, which then afterwards was used to elicit targeted neutralizing antibody responses. So I want to briefly introduce you what protein design actually is and what it means in practical terms. So when a protein is translated at the ribosome, just as is happening all the time in our cells, this linear chain of amino acids folds into a three-dimensional structure. So this is what is referred to as protein folding. And the three-dimensional structure these proteins adopt are typically the lowest energy state that is available to that sequence. Now, protein design can be considered the opposite. And what we do here is we define a three-dimensional structure that we want to obtain, and then we search for a sequence that can fold into this structure. And to do this, we use computational techniques. There is a protein design software called Rosetta that we use mostly to explore basically the sequence space and the structures that the sequences that can fold into the particular structure that we want. So you can basically define protein design as a search problem where you're searching for a sequence that can fold into a given structure. So we wanted to use now protein design to develop a multi-site cocktail vaccine, and this is what I'm talking about today. So again, you see on the left the pre-fusion RSVF protein, and 3 epitopes are highlighted here. There is the site 0 at the apex. Site 0 is targeted by the antibody D25 or nirsevimab. Then site 2, which is the target of palivizumab, and then in orange shown site 4. Site 4 is an epitope that is conserved between different viruses and so targeted by cross-neutralizing antibodies. So one of the challenges for us as protein designers is the structural complexity that in particular the site 0 has. So site 0 consists of 2 different segments. So you see a helical segment and a loop segment. And as I will show you in the next couple of slides, stabilizing an epitope that consists of more than 1 segment is not very trivial and really needs state-of-the-art protein design methods. So to cope with this problem, what we've done is we isolated, so excised the site 0 from the native context, so from the viral fusion protein. And then we designed a protein that can stabilize this epitope. And to do this, we developed a new protein design algorithm that is called TopoBuilder. And so what this TopoBuilder does is it builds customized protein topologies that can stabilize such complex motifs. So it basically works in 3 steps. So in the first step, we define a protein topology in the 2D space. So what we have here in purple depicted is the epitope simplified as a 2D representation, a circle and a cross. And then in a 2D space, we place an additional helix, helical secondary structure element that we then expand into the 3D space. So now we're going over into the 3-dimensional space where we generate a sketch, a very low resolution sketch of how this protein ultimately should look like. And then lastly, we perform in the computer a protein folding and sequence design simulation, which we do with the help of Rosetta. So I should tell you that protein design or computational protein design is really not a perfect process. And instead, what we heavily rely on is experimental screening and also sometimes experimental optimization. So what you see here is that we start by computationally modeling 10 to the 4 sequences. So we generate many sequences and many computational models in the order of 10 or 20,000. And then we use a method that is called yeast surface display. So this yeast surface display is a very well-established technique from the directed evolution community. And what you do with this method is you can display the proteins that you designed on the surface of yeast. And then you can apply different selection pressures and use flow cytometry to isolate sequences which have the desired properties. And here in particular, we were screening for 2 different conditions. So one of them is the stability of this protein in terms of folding and also thermostability. The other condition is antibody binding. And the reason why we want the antibodies to bind is because the better the known neutralizing antibodies can bind, the better we are actually mimicking the epitope in this computationally designed protein. And so after the screening process, what we do is we recombinantly express some of these proteins and then biophysically characterize them. And this is typically a handful of sequences that we characterize. So to cut a long story short, so what I'm showing you here is designs for site 0 and also for site 4, because the site 2 immunogen was published before. And what you're seeing here is that upon recombinant expression and biophysical characterization, these proteins are extremely thermostable, so up to 80 degrees, which may be important whenever these proteins would be used in regions of the world where they could not be refrigerated all the time. And then they bind with extremely high affinity, so with picomolar affinity to the target neutralizing antibodies, which from a protein design perspective was really a big success. So then, since we are mostly doing computational design, what we ultimately want to do is we want to check if an experimentally determined structure by a structural biologist actually superimposes with the model that we generated to actually prove that our computational model is correct. And this is what you see here. This worked out very nicely in this case. You see in gray the computational model, so what we designed and predicted in the computer. And then in orange or in purple on the right side, you see what the experimental crystal structure was determined. And these two structures really superimpose very, very accurately, meaning that the protein design approach that we applied has worked very well and has generated accurately what we intended them to be. So then when you zoom in, and this perhaps is most important, is that you can see that even on the side chain level, we are mimicking exactly how these epitopes look like in prefusion RSVF. So overall, we have a sub-angstrom mimicry of these neutralization epitopes in these de novo designed proteins. So I just want to summarize this first part before I then go on and show you what these proteins can actually do in vivo. So we developed this de novo template building algorithm called TopoBuilder that enables you to build tailored proteins for structurally complex motifs. In general, in this case, it was neutralization epitopes, but could be applied to other motifs as well. So the proteins that we designed this way are thermostable and bind with extremely high affinity to their target neutralizing antibodies. And then finally, crystal structures confirm that the computational models were actually accurate. So now in the second half of my talk, what I will show you is the immunization studies that we've done and where we aim now to use these designed proteins to actually elicit neutralizing antibodies targeting 3 different sites. So one of them, the site 0, second site 2, and then the site 4. So to do this, what we did is we multimerized these proteins on protein-based nanoparticles. The reason for this is that these nanoparticles increase the immunogenicity of these proteins, and they also have a certain size, which makes it easier for the immune system to recognize them, and also this multimeric effect helps to stimulate the B cells. So, and then we mix these 3 immunogens together, we refer to it as Trivax, and then we tested this in animals. So we started by mouse immunizations, and we basically had 3 groups. The first group, saw Trivax 3 times, so every 21 days, every 3 weeks. The second group as a control got pre-fusion RSVF, so the protein that was developed in Barney Graham's lab at the NIH. And then in a third group that I will later come back to is where we primed animals with pre-fusion RSVF and then boosted them with Trivax. What you see then is the pre-fusion RSVF titer after 2 weeks after the last immunization. And you see that Trivax actually elicits serum levels of antibodies that cross-react with the viral protein, which by itself is quite a success, because we are only presenting a small subset of epitopes compared to the full viral protein. Obviously, pre-fusion RSVF, the titers are much higher compared to what Trivax can elicit. But what was very striking to us is that actually by just presenting these 3 epitopes, we can actually get RSV neutralizing activity in these mice sera in 6 out of 10 mice. So what I'm showing you here is measurement of RSV neutralization using a standard in vitro assay. Again, compared to pre-fusion RSVF, currently the leading immunogen, these neutralizing titers are inferior because also we are presenting less epitopes than pre-fusion RSVF. So what I would like you to remember from here is that if you think about the different scales, it is actually quite striking to get any neutralizing activity. So starting from this virus, where on the surface is the viral fusion protein, we isolated these neutralizing epitopes and then used computers to create these synthetic proteins, which then actually re-elicited antibodies that can neutralize the virus, which is a very high bar for a protein that ultimately comes out of a computer. So we then took this forward and went to immunize non-human primates in collaboration with a Canadian university. And these primates were immunized 4 times every 28 days. And what you can see again is that we elicit RSVF cross-reactive serum titers. And this time we can also prove that in fact these serum titers are directed against all 3 sites. So we have site 0 specific antibodies, site 2 and site 4. And across all 7 animals that were immunized, we can actually say that is a very balanced response, where we have a relatively predictable amount of site 0, site 2, and site 4 antibodies. And then, what in primates was even more striking compared to mice is that after only a single boosting immunization, so at day 35, we actually have RSV neutralizing serum levels in 6 out of 7 animals, which is what you see in this plot. So then we also went on in collaboration with Yuqing Li at the University of Maryland to isolate monoclonal antibodies, so from the memory B cells. And here what we found is we found antibodies against site 0 and site 2 that bind pre-fusion RSVF, that do not bind to epitope-specific knockout mutants, so they are site-specific. And then we also tested them in an in vitro neutralization assay and can actually confirm that both monoclonal antibodies are neutralizing for RSV in vitro. Perhaps most interesting is the antibody that we found against site 2, because we realized that it's actually about 10 times more potent than palivizumab. We then also solved a crystal structure again of the antibody that we elicited against site 2 in complex with the immunogen, and we can actually show again that is basically site-specific and that is compatible with binding to pre-fusion RSVF. So as my last point that I want to make, where I think that these immunogens are also very interesting, is on the idea of actually boosting site-specific antibodies. So this obviously is a problem in RSV, but also in other pathogens where basically the population has experienced several infections before and therefore has pre-existing immunity. And the question that we asked is, can we use our Trivax immunogens to selectively boost site-specific antibodies after, so to say, a broad subset of B cells has been primed? So, and to mimic this, we primed these animals with pre-fusion RSVF and then aim to boost site-specific antibodies. So the outcome of this experiment is shown in this slide, where now I just want you to focus on group 2, so the gray group, which has seen RSVF 3 times, and then the heterologous prime boost group, which was primed with RSVF and then boosted with Trivax. And then what we found is that actually the fraction of site 0 specific antibodies could be increased by boosting with Trivax, so the blue versus the gray. The same is true for site 2, so we can boost site 2 specific antibodies and also site 4 specific antibodies. And this is also then reflected if we compute the so-called antibody quality, which is a very common measure for vaccine success, where you basically compute a ratio between antibodies that bind and those that neutralize. And the lower this ratio, the higher the quality of this antibody response is. And now if we compare animals that have seen 3 times RSVF versus those which were primed and then boosted with Trivax, we can actually lower, so improve this ratio of antibody of binding to neutralization. So to summarize the second part, what I've shown you is that this trivalent cocktail of computationally designed antigens enables you to induce RSV neutralizing serum levels directed against 3 different epitopes in this case. Then we could show that monoclonal antibodies that we could isolate from these animals after they were vaccinated are epitope specific and are able to neutralize RSV. And then lastly, we can show that after priming with prefusion RSVF, Trivax allows you to boost site-specific antibodies, and this leads to an improved quality of the antibody response. So just to finish, so basically this is a graphical abstract of this work. So starting from the epitopes that we isolated, we developed this TopoBuilder protein design algorithm, which then generated stable immunogens, which could elicit potent neutralizing antibodies against RSV. And we think that this, especially the protein design tool, has exciting applications also beyond the design of immunogens. So basically the selling point of this algorithm is that it can deal with very complex functional motifs or very complex neutralization epitopes. And for the vaccine design community, we think that this approach keeps being exciting for pathogens that require fine control over the antibody specificities that need to be induced to confer broad and potent protection. And here, other examples could, for example, be influenza or dengue. These proteins also are very exciting probes for monoclonal antibody discovery. And lately, we have also transformed these proteins into diagnostic tools that allow you to detect epitope-specific antibodies in serum. And with this, I would like to thank all of you first for your attention, and then I would like again to thank the RSVNet Foundation for organizing this seminar, our funding sources, and then in particular Bruno Correa, the PI, and Che Yang, so both of them you see here in pictures, that worked with me on this project, then facilities at the EPFL, and a number of very important collaborators. And here I also want to point out Sabine Riffaut and her team from the INRA in France, who helped us a lot with the nanoparticles and also with some immunization studies. Then collaborators on the structural biology side, Yuqing Li and her team for the antibody isolation, and then also Martin Merks and his team from the TUE for working with us on biosensors to transform them into diagnostic tools. Thank you very much, and I'm happy to take questions.
Thank you very much, Fabian. That was spectacular. Very, very nice. Also, congratulations to the others of the team. I can see that others are actually in the meeting. I'm just want to be sure that can you hear me?
Mm-hmm.
Yes. Okay. Fantastic. So again, Fabian, thank you very much. The people on this meeting, feel free to ask questions. At the moment, it is not possible to directly interact with Fabian for technical reasons. I think you can understand how difficult that is with such a large audience. So you can ask your questions. Some of you have started to do that by the Q&A button, and then I can ask those questions to Fabian. So I'll start off, Fabian, after my congratulations, with a rather technical question. I think what you have shown is that after priming with a pre-F vaccine, that boosting increases the ratio between neutralizing versus binding non-neutralizing antibodies. Is that a sort of a fine-tuning of the immune response to neutralization? And do you think that this is something that can be used more broadly also in the development of other vaccines, that you can use this TopoBuilder epitope-specific way of vaccination to fine-tune a broader primed immune response? Over.
Yeah. So that's exactly basically the idea that we are working on. And so I do think that the reason why we can improve this binding to neutralizing antibody responses is because we are really boosting basically antibodies against epitopes which are very sensitive to neutralization, right? Now, as you might also have seen in one of the slides, it always depends on the exact readout that you have. So if your only readout is overall neutralizing activity, then repeated boosting immunizations with prefusion RSVF still give you slightly higher neutralizing titers than just one and then boosting with Trivax. But if you're going after basically site-specific neutralization, then this approach seems to be superior. But we're also still working on further validating this in other animal models.
Thank you. Thank you very much. So if you would, if you would use this system in non-naive humans, how would that work?
Yeah, so I mean, the answer is obviously we haven't tested it, and this is something that we're very interested in doing, and actually trying to boost antibodies not after a prefusion RSVF immunization, but after actually infection. So basically to really boost non-naive individuals. And here we're planning to do it in a non-human primate study, but so far this hasn't happened yet. But this is something that we're interested in.
Thank you. So I have some delays. So the sound is fantastic, and the video at my side is there is some delay. So I hope you don't have that at home. Fabian, I get a lot of enthusiastic responses in my inbox, so I'll share all of them later on, or you can have a look at them while I'm talking. So one of the questions from the audience is, what about Th1, Th2 immune response, or beneficial versus a more dangerous immune response? Did you have a chance to look at a Th1, Th2 bias in the non-human primates, I assume?
Yeah, it's a great question, and the answer is we have not looked at it. So it's something that we would like to do, but I basically have no answer to this question at this point, because we just haven't analyzed any T-cell related response.
So basically upfront, using this technology, you cannot predict whether there's going to be a Th. It's not a system that biases towards one or the other. Is that correct?
Yeah, no, it's not. I mean, we are basically agnostic of T cells at this point, because we just basically read out B cell responses at this point. But it's definitely something that we have to do, and that we're also very interested in, especially if someone is willing to collaborate on this subject.
So a very obvious question is, where are you in the development of this vaccine? What is the current status of the vaccine development, preclinical, clinical? What is your ambition? I think people are very eager to learn where this is going.
Yeah. So, I mean, as you've seen, we have done a first non-human primate study, and we are currently looking into additional non-human primate study with an optimized formulation of this vaccine. So, well, as you can probably imagine, this class of immunogens is entirely new. And we believe that there is a lot of room for actually optimizing the magnitude of the immune response. So what we see is in the primate study is that after a single boosting immunization, which I think is acceptable, 6 out of 7 animals actually neutralize RSV. But we would like to see higher neutralizing titers, which reach a level that is more close to what prefusion RSVF currently can do. And we believe that there is a lot of room for optimizing nanoparticles and also adjuvants and formulation conditions. So this is something we're working on currently. And again, if someone is willing to collaborate on this subject, we would be happy to. But currently our plan is to test an optimized formulation again in non-human primates and then think about the next steps.
So I can see that some of the people are anxious to see some human studies because they are asking, what is your target population? Naive neonates or pregnant women? I think it could match with the maternal vaccine program, but it would make things much more complicated, specifically in this, let's say, highly protected pregnancy. So what is, if you would go to humans at the end of the day for your target population, what do you think it would be most useful for?
Yeah, so I think there is arguments for different target populations. One thing that we're excited about is basically pregnant women, vaccinating pregnant women is something that is currently being tried, and we think that it would be beneficial to vaccinate pregnant women in a way that the antibodies that get transferred do not interfere with actually the antibodies that babies can make. And it was shown in a study, I believe by Laura Walker and others, that essentially babies can mostly make antibodies that recognize RSVF on the site. And we believe that one approach would basically be to boost site-specific antibodies in the mothers that would not interfere with the baby's immune response at an early stage. So that's one approach. If babies ultimately would be an option, I think it's a very complicated subject at which age you can actually vaccinate babies, and if this vaccine would be, has the right level of potency and safety, we don't know yet.
So, do you have data after 1, 2, 3 doses with Trivax? How much can you boost the system?
Yeah. So what we're currently seeing, and currently I mean with the current formulation that we have, is that in fact after the second immunization, titers do not go up anymore. So we're reaching a plateau basically after 2 immunizations. So that's basically what we have currently.
Is there any evidence of cross? So you're seeing, Fabian, you're doing fantastic. The questions are coming now rapidly, and I'm shooting all those questions to you. So I feel very, I don't know, it's not very polite, just shooting all those questions at you, but you're doing a fantastic job. I'm sure people agree to that. So is there any evidence of cross-reactivity to of the immune response to irrelevant human structures? I think you can understand that people are always concerned about safety when you're developing a new methodology for vaccine development. So cross-reactivity to non-related human targets.
Yeah, it's a great question. So in principle, these immunogens should not have any at least predictable cross-reactivity. I mean, first of all, mostly they consist of the epitope, and the rest of the protein is completely de novo. So it's basically artificial, and in terms of sequence, and also in terms of structure, but mostly in terms of sequence, they are extremely far from any known human protein.
Thank you. So if you look at all the epitopes in PreF, is there any reason that one epitope would work differently from the others in your TopoBuilder capacity. I'm not sure I fully understood the question, Sabine, so feel free to refine it through the Q&A. But can you speak to that, Fabian? The different epitopes, do they all work equally well, if you could predict?
Yeah, I think I see Sabine's question here. It's a very interesting question and really a key question. So let's say that epitopes which consist of like many segments, and here I'm speaking about 5 or even more segments, they would still be very challenging to actually design scaffolds for. Now, what the TopoBuilder has allowed you is to actually go from very simple structures, which, for example, the site 2 is a very simple epitope, structurally speaking, because it's a regular helical segment, to more complex epitopes. And more complex means, you know, any sort of beta strand is more complex because it's much harder to stabilize, and then also multi-segment epitopes. So we are currently working on additional RSVF epitopes as well, which are even more complicated than what we've been working on right now. Now, inherently, there will be a limit whenever you go beyond 5 segments, I believe. But yeah, at this point, you know, I think in terms of the RSV epitopes that are known, we could probably cover most of them using the TopoBuilder.
Thank you. Thank you. Thank you for explaining that so carefully. Feel free, Sabine, to do a follow-up question. So Sonny was wondering whether you have challenged the monkeys with both subtype A and B. And is there any evidence or any reason to assume that your immune response to Trivax works differently for A versus B?
Yeah, also a great question. So first of all, no, we did not do a challenging study yet, but it's something that we're planning to do. Now, subtype A versus B, I mean, A is by far the most dominant strain, of course. Now, there is a small difference in the sequence of the antigenic site 0 of type A versus type B. And we think, although we haven't done it yet, is that we would have to adapt the site 0 immunogen to also be compatible with basically the sequence that strain B has. But that is the same problem that pre-fusion RSVF also has.
Thank you. So if we circle back to this question of refinement or focusing the immune response to the protective epitopes that your Trivax is appearing to do, you mentioned that it could be a booster strategy. Have you considered the opposite of priming with Trivax and then boosting with Pre-F? And what would happen if you would do that? Would you limit the response to a much broader Pre-F vaccine if you would prime first in a naive baby, for example, with Trivax?
Yeah, that's also a great question. And so our initial idea was to not do it simply because, you know, there is pre-existing immunity, and therefore we thought that the other way around makes more sense. Nevertheless, we haven't fully done this experiment yet. Yeah, that's basically the answer. We haven't really done the experiment yet. I think, you know, it would be an interesting scenario, especially for the infants. There is one thing that we have observed throughout the years, and this is that whenever we prime naive animals with Trivax, is that we do get a lot of antibodies that are basically not that bind to the epitope in the scaffold, but that are incompatible with binding pre-fusion RSVF. Okay, and we think that therefore it is a better strategy to actually prime the right subset of B cells with pre-fusion F. And then from that prime pool that is already compatible with Pre-F, boost those which target these sites. So we think the other way around is a better strategy, but yeah, we're also working on trying it the other way around.
So I got a question about similarity or differences between the epitopes that you're using for your Trivax. How similar or different are they? I have assumed that they are very different and non-overlapping. Can you speak to that a little bit, Fabian?
So I'm not sure I fully understand the question. So the epitopes that we selected, structurally speaking, are far apart. So basically, we selected them because site 0, site 2, and site 4 are non-overlapping epitopes. So antibodies can actually bind to all 3 sites simultaneously.
Yeah.
Yeah, I don't know if this answers the question.
So we should see this as 3 independent immunizations, basically.
So you should see it as 3 different immunogens, but that then are delivered as a cocktail vaccine, right, where all 3 immunogens are mixed together.
Yeah. Are there any regulatory hurdles for 3 immunogens versus 1 protein-based vaccine? Have you looked into the regulatory hurdles that might occur using combination immunogens?
Yeah, no, we have not looked into regulatory hurdles simply because we're not at this point yet. It is true that manufacturing 3 different immunogens may be a challenge, but we also think that there is a solution if if this proves to be better than an existing solution.
Thank you. It seems that we're going to get closer to the end of this session. Thank you very much, Fabian. The RSV world is very much used to not to be at the front page. Influenza is always our #1 competitor. Nowadays, we are even lower down the list. We are at least #3 following COVID, SARS-CoV-2, and influenza. So what prospects do you have using this technology based on what you've seen in your pretty successful RSV Trivax experiment, that you can use your TopoBuilder for viruses that are much more famous, maybe not so important, but definitely much more famous than RSV? Just give us a little bit. I understand that this is a very wild shot, but I'm sure it's in the back of the mind of many. Can we use it for what can we use it for? Any ideas? Ebola? Give us some ideas.
Yeah. Well, I mean, in fact, we are working on applying this strategy for influenza as well, because we think that there is a lot of potential as well to boost antibodies which are in the stem region and that are known to confer much broader protection than all these head antibodies. Now, in order to really apply this strategy in a way that it makes sense, what you need to know is the actual epitopes, and you need to have structural evidence and structural information about these evidence, which in the case of SARS-CoV-2, I mean, they are coming out, these studies, but it's still an early stage. And what we have really benefited here is the decades of work that other people have done to actually structurally characterize what antibodies are the good ones to neutralize RSV. Yeah. So besides influenza, I also think that there are exciting applications in the case of dengue, where there is different subtypes and only few epitopes which are actually conserved among these different strains. So that's basically another application that we are envisioning. So
I have another question coming in that relates basically to the immune response that you're seeing currently in your studies, related to the use of adjuvants. Do you know what part of the immune system is used in your Trivax model, and can it be enhanced or potentiated using adjuvants?
Yeah, so in fact, I probably have forgotten to mention that these immunogens, they were formulated with an aluminium adjuvant. Just the same, basically, that is also used in pre-fusion RSVF clinical trials and in many licensed vaccines already. So this was formulated with an aluminium adjuvant. Okay, thank you very much. There's an excellent suggestion that in your future experiment, you might consider using clinical isolates, Fabian, in order to make it as useful as possible. People would like to see you use clinical isolates and not just very, very old lab strains.
Are you considering that?
It's a great suggestion. We would love to collaborate if someone, you know, is an expert on these clinical isolates and has them around, basically, for sure. Yeah.
Thank you. There are a lot of folks on this call that actually have access to that, so I think this will be followed up, I'm sure. I think we can conclude that this was a. I think there are no more questions. I think we can conclude that this was a successful first webinar from RSVNet. But I'm very curious to hear your feedback. So after this meeting, please stay tuned. You will receive a request for let me see. You will receive a request for feedback, evaluation, maybe things you would like to see in the next meeting, or things that a topic that you would like to see replaced, or a co-host that you would like to see for the next meeting. We are learning, so be frank. Fabian has agreed that we can share his presentation, so it will stay online for those of you who joined later. If you didn't see everything, then it's going to be available hopefully from tomorrow onwards or so. Very soon we will make this available. And I'm looking at Amber. Is there anything else that we need to discuss, or can we close? Okay, I get a signal from the real boss here who's telling me what to do. She is whispering in my ear everything that I'm saying now. So thank you all very much for joining. And in particular, I know if it would be possible, it would be a big applause to you, Fabian, for doing a fantastic, really fantastic, impressive, and all the superlatives that I heard in the Q&A for kickoff of the webinar series of RSVNet. Enjoy the day, and I hope to see you soon. Bye, everyone.